Action on the molecular level, the sliding of protein filaments, is responsible for muscle contraction. When a motor neuron stimulates a muscle fiber, overlapping thick and thin filaments slide along one another and sarcomeres shorten. The combined shortening of many sarcomeres in many muscle fibers results in contraction of the whole muscle. Contraction only shortens the sarcomeres. It does not change the lengths of the thick and thin filaments. What causes the thin filaments to slide across the thick filaments? Thick filaments in a muscle fiber are made of many molecules of the protein myosin. Each myosin molecule has a fibrous tail region and a globular head. For clarity, only one head is shown here. Thin filaments are twisted chains made of the globular protein actin. When a nerve impulse arrives at a muscle fiber, calcium ions trigger muscle contraction and ATP provides the energy. The head of a myosin molecule can attach to the actin of a thin filament. When an ATP molecule binds to the myosin head, it detaches from the actin filament. The ATP molecule is hydrolyzed into ADP and a phosphate group, which remain bound to the myosin head. This causes the myosin head to change position, storing energy that is later used in contraction. With the myosin head in its high energy position, the muscle fiber is ready to contract. A nerve impulse stimulates the muscle cell. This triggers the sarcoplasmic reticulum to release stored calcium ions. Calcium ions interact with other proteins on the thin filament, opening up binding sites on the actin and making it possible for myosin heads to bind to the actin. As ADP and phosphate are released, the myosin head bends. This pulls on the thin filament, causing the thin filament to slide toward the center of the sarcomere. More ATP binds to the myosin head and the whole process repeats. The myosin head continues to move and pull on the actin filament and the muscle contracts as long as calcium ions are available to open up actin binding sites until the muscle fiber is fully contracted. When the motor neuron stops sending impulses to the muscle, calcium is pumped out of the cytoplasm and back into the sarcoplasmic reticulum. Because calcium ions are no longer available, the binding sites on actin become covered again, and so the myosin head cannot bind to the actin filament. As all of the thin filaments slide back to their relaxed positions, the entire muscle relaxes. Only one myosin head is shown here, but a typical thick filament has hundreds of heads pulling against several surrounding thin filaments at once. Each myosin head is able to bind and unbind about 5 times per second. The heads do not work in synchrony. Some are bound to thin filaments, while others are reaching for new binding sites. Smooth contraction of a muscle results from the simultaneous combined pulling and sliding of millions of thick and thin filaments in thousands of muscle fibers.